Welcome to the UMass Core Facilities Seminar Series. Today we are hosting the Light Microscopy Facility with Director Jim Chambers. Today's topic is Rethinking the Impossible, Powerful and Cutting-Edge Imaging Technology, Bringing Your Experiments to Reality. We will be hearing testimonials from three researchers, Pat Wadsworth in biology, Carline Fermino Do Rosario from MCB, and Joseph DiPietro from Nikon. I am Lisa Korpieski, the Director of Communications and Marketing here at the Institute for Applied Life Sciences. We hope that with these bi-weekly seminars, you will discover what great resources that the centralized UMass Amherst core facilities offer to our campus community, the New England region, and beyond. Just a few housekeeping items. The seminar is being recorded, as you just heard. All replays of the seminars in this series can be found on our website. I will put a link in the chat. Please save your Q&A till the end of the presentation. During that time, I welcome you to unmute yourself and ask your question. Next, I would like to introduce you to Andrew Bernard, the Director of our UMass Amherst core facilities. Andrew?
Thanks, Lisa. Good afternoon, everyone. Thank you for joining us for the 6th Fall 2021 Core Facility Seminar. As Lisa mentioned, my name is Andrew Bernard, and I'm the Director of Centralized Cores here at UMass Amherst. If you've joined us before, you've heard about our state-of-the-art labs, instrumentation, services, and most importantly, our world-class experts who are helping researchers at UMass and beyond drive discovery and pursue scientific endeavors across all disciplines. I'm grateful and honored to be able to share these facilities as broadly as possible. Core facilities are open to anyone from students through senior scientists, regardless of your affiliation, including researchers from other academic institutions and commercial organizations. In order to maintain these operations at affordable rates for our users, the core facilities receive a significant investment from the university. In addition to this investment, the existence of cores depends in part on proper acknowledgement and publications. It's an important metric of value for most core facilities that enables them to obtain additional support so they can continue to provide essential services in the most comprehensive ways possible. It also helps our core personnel, many who do not conduct independent research, to advance in their careers, adding to the overall health and standing of the core facility. You can learn more about suggested authorship and acknowledgement guidelines on our websites or via the ABRF. I'll put a link in the chat in a few after I wrap up. To help access our cores, there are several funding opportunities. For our IELTS investigators, there are core credits and center seed funding available. And for those who are external to UMass, the Mass Innovation Voucher Program subsidizes usage for small companies based in Massachusetts at up to 75%. The program has awarded more than 850 vouchers, representing more than $15 million in project costs across all 5 UMass campuses over the last several years. We're here to be your partner. We hope to expand your research capabilities and bring you productivity. Please feel free to reach out to me or any of the core facility directors if you have any questions or would like to learn more about how you can engage with us. With that, I'll turn it over to Dr. James Chambers, Director of the Nikon Center of Excellence, Light Microscopy Core, who will tell you a bit more about the facility and introduce our guest speakers. Go ahead, Jim.
All right, thanks, Andrew. Thanks, Lisa. So I'm going to keep this quick because I know anyone who's here is actually here, not for me, but here to learn about some of the science we're doing here. So what the core facility does here, the Light Microscopy Core Facility, is we provide access to high-end imaging setups. Everything we have is state-of-the-art. Our rates are extremely economical. And like Andrew said, we're a Nikon Center of Excellence. What that lets us do is really partner very close with the Nikon, for instance, having Joe here today, on all things from basic training to cutting edge new technologies. It's a really great relationship. So how can we help you? We have kind of 3 phases of help. So from before your experiment, we can meet with you to consult to make sure you're going down the right path when it comes to microscopy, to get the data that you need to answer your questions. During your experiments, we can check quality, make sure you're getting the right data. And after, we help people with analysis pipelines quite a bit. All of the microscopes, for the most part, run the same software. So switching different technologies or modalities is very simple in our core facility. Now, as far as the technology we offer, it pretty much covers the gamut of light microscopy. Our microscopes are all self-operated. We do offer imaging service. We place a high premium on training. So one-on-one training is done for all users. And that is everything from people who can't even point to an objective all the way to, advanced scientists who want to kind of take it a little further. We offer extensive help that's available 24/7 on Microsoft Teams. There's a bit.ly link here if you want to join. The software is always kept up to date by Nikon. And like I said, image analysis is kind of one of the key things we spend a lot of time on, and it is a big focus, pun intended, of the light microscopy facility. So we currently have 14 microscopes. I'll just blow through them really quick here. 5 scanning confocals, 3 turf, 2 spinning discs, 1 sim, a high content stereoscope, single molecule localization microscope, laser capture microdissection, a simple walk-up microscope, as well as a patch clamp rig. Now you'll notice that adds up to more than 14. Many of our microscopes are kind of 2 or 3 microscopes in one. So some of the capabilities we can do, this is not an exhaustive list: thick Z-stacks, resonance scanning, multi-photon, live animal work, multiplexed imaging, automated imaging, super resolution, as Joe will talk about a little bit, FRAP and photoactivation, FCS, FLIM, as well as algorithms including deconvolution, artificial intelligence, and machine learning. Here's my contact info, as well as Maya, who's a light microscope staff member. And with that, I will turn it over to Pat and Carline.
Yeah, all set? Yeah. Okay, can you hear me? Ooh, you might have Okay, so thank you very much to Lisa and Andrew and Jim for providing us this opportunity to tell you a little bit about the wonderful things we get to do with all the wonderful microscopes over in the facility. And so I want to start by just trying to impress you a little bit with the power of live cell imaging. So these 3 videos that I'm going to play for you are examples of how you can gain insight into the localization of components in complex cellular structures. And so the structure that we look at is this mitotic spindle in a relatively suitable for microscopy kind of cell, which lies very flat. And you can see that we can tag with GFP microtubules, proteins that are at the centromeric, where the chromosomes are connected to the spindle, and also proteins at the tips of the microtubules. But as beautiful as these movies are, and they're all taken on a spinning disc Nikon microscope, it's also important to probe a little bit more deeply and to try to understand the dynamic behavior of individual protein components within the mitotic cell. And so I just wanted to remind you of this incredible revolution in live cell imaging brought about by the discovery and application of GFP. And so you can see here the beautiful beta barrel of the original GFP molecule. And GFP has a wonderful advantage, and it's very readily photo bleached. That can also be a negative, but you can take advantage of that and bleach GFP in particular regions of the cell and then study its recovery. Over the years, a whole host of either modified GFP variants or other fluorophores have been discovered and utilized. So there's a huge realm of different tags that you can add to your protein of interest. One probe that we've used a lot and that Carline is going to tell you about, she's going to tell you about FRAP and about photoactivation. And these are a particular type of GFP shown here on the side. It's actually non-fluorescent, it's dark due to a mutation in the amino acid sequence of GFP. However, when UV light is applied to the sample that has a photoactivatable GFP, it becomes green, and so then you can see it. So this allows you to activate just a subset of the molecules in the cell and then study their behavior. Likewise, things I won't be talking about is there are photoconvertibles, dyes that are one color, here green for EOS. You shine light on it and it becomes red. So again, you can mark different populations of molecules within the cell. And when we get to our super resolution part of the program, we might hear about switchables, which can blink back and forth and switch between light and dark. Before I hand it over to Carline, who's really going to go into some experimental data using both photo bleaching of GFP and photo activation of GFP, and explaining to you how you collect that data and how you analyze it. As Jim said, analysis is really important. I wanted to put a plug in for two super resolution techniques. I don't think I'm stepping on Joseph's toes because he's going to talk about POM. One is expansion microscopy. These cells were imaged on an A1R Nikon point scanning confocal microscope. And this technique is just super cool. You basically fix your cells. They can be GFP expressing, or you can tag them with antibodies. And basically you embed them in a gel. And this gel, upon addition of water, can swell. And when it swells, you basically expand the sample, and thus you gain additional resolution. And I think you can appreciate a pre-expansion mitotic spindle shown here on the left, and a spindle post-expansion at the same magnification, and two views where you can, basically see every single microtubule in the cell. So the imaging is really beautiful. You can also take a full movie, Z-stack. And again, this is where analysis comes in handy, because you can actually start to look in 3 dimensions at such a volume that's collected. And I have to thank the group of Ed Boyden at MIT, because they were really helpful in getting us started on expansion microscopy. And finally, before I turn to Carline, another technique in the center that we have used is the Nikon SIM microscope, which is structured illumination. And again, just a brief overview to hopefully get you excited about the things that can happen over in the imaging facility. This is a different kind of mitotic spindle, comes from a amoeba called Naglaria. It's a collaboration with Lil Fritz Leyland. And here is a spinning disk image of that spindle. And then here are examples of spindles in cells at the same stage of mitosis. And I think you can appreciate the detail that you can now see within this spindle. These are microtubules again. So are these, but they cannot be individually resolved. And these spindles are like little barrels. And so you can look at them in cross section. And here's a cross section of such a spindle with a spinning disk microscope. So this is the spinning disk in the facility. And here it is, similar image taken with the structured illumination. So to just conclude this very brief introduction before Carline starts, there's a tremendous amount of information that can be gained by simply observing cells alive on the microscope, but also from probing them with these techniques of photoactivation and photobleaching, which you'll hear about from Carline. And also super resolution offers a lot of advantages, and we're really happy that it's available to us. So I'm going to pass it over now to Carline. We're working off the same PowerPoint. And so here she's going to tell you about the work she's been doing in my lab and with tons of help from the Nikon Center.
Can I move? It's not working.
Not working?
No, I was trying to move this because I think it might be blocking some of the slides. Okay. Perfect. Okay. Hi, everyone. I'm Carline. And again, thank you to Dr. Chambers and the Isles for the opportunity. And I'm going to be talking a little bit about some of the exciting research I've been doing. So in this video, you could see a cell where all the microtubules are labeled green as it's going through cell division. And so the Wadsworth lab is a cell biology lab that studies the mitotic spindle. So one thing I want to highlight that you could see in this video is that this structure is highly, highly dynamic. And you could see that from its transition to metaphase. Here you see kinetochore fibers that help align the chromosomes in the equator of the cell. And as the cell transitions from metaphase to anaphase, it assembles all different types of structures that help further separate and elongate the spindle so that they could divide into two daughter cells. One particular structure I want everyone to focus on that I'm going to be talking in more detail is this midzone, or also called central spindling, which are the microtubules in between the two set of chromosomes. So we've been using two techniques to study how dynamic this structure is. And the first one I'm going to be covering is fluorescent recovery after photobleaching, or also known as FRAP. So you could start with a fluorescent structure, as you could see here, pre-bleach, where you see all the fluorescence. You could choose an area of interest. So here I'm showing it in this circle, purple circle. And after exposure to light, then it loses its fluorescence. And then you could watch for the recovery, or how fast there's exchange of bleached and unbleached molecules that within that region that was photobleached. And before I show a graph, or the ideal graph of this experiment, I just want to highlight that within the region that you photobleach, not all molecules will exchange, and so not all molecules will exchange between fluorescent and bleached ones. So here is an ideal FRAP data, and so as you could see, here is the pre-bleached intensity, and then you bleach it, and this is the recovery curve of an example FRAP. And again, you could see that it doesn't really recover all the way to the initial intensity, because again, not all the molecules are going to exchange between the bleached and fluorescent ones. So this is just how an ideal data for FRAP would look. And then you could, based on this recovery curve, you could get a half time that would tell you how fast that recovery is happening. And then in here, in the second graph, is just an example of an actual FRAP data done, and where you could see the boxes where the photobleaching area, or specific area of interest, and then you bleach it, and then this is the recovery curve for this particular example. One, which I should have highlighted before I showed and now, sorry, I'm going to show some examples of FRAP that we've done in lab. So here you could see a video of a FRAP done in a anaphase cell. And one thing I want to highlight is that it might be a little hard to see in this movie. Sorry, it should have been moved. Okay. How fast this recovery is happening. So then you could appreciate that in the stills. So this is before bleaching, and this is the area that was bleached. And then here you could see the recovery of fluorescence within that specific area. And we did it again in a much later cell, and you could see again, this is before the bleach, and then this is the area that we bleach and the recovery. One thing that you might observe is that there was faster recovery in the previous cell compared to the cell. And again, that could be because the previous structures are more dynamic. Before I go on and talk about photoactivation, I just want to highlight that it's a fantastic technique, but it might be a little hard to see that recovery, especially with dynamic structures, when you have every fluorescent microtubule labeled, as you saw on the previous cell. So then to combat this, we decided to go with photoactivation of fluorescence. So the technique works a little different than FRAP. So you start, as Pat mentioned earlier, you start with the dark background, and then you choose an area of interest and you photoactivate it upon UV light exposure. So all of the molecules of interest within that region turn green, and then you could observe for the dissipation of that fluorescence over time. And before I go and show the amazing videos we've been able to collect at the Light Microscopy Center, I just want to say that before you proceed to doing experiments, there has to be some characterization of the cells. So this is a cell line that we created in lab. And before we moved on to doing experiments, we want to make sure that, one, all the microtubules are activatable within the cell. As you could see here, this is pre-activation and post-activation. And then secondly, we are a lab that studies the mitotic spindle. So it's crucial that our cells are able to progress through mitosis and actually complete so that we can study all the structures within that, within the spindle. So here is pre-activation. Here's a metaphase spindle, and then the cell actually it progresses through cytokinesis and it's able to divide and it's healthy. So now I'm showing two examples of photoactivations done at the center. So you could appreciate that it starts from a dark background, and then upon activation, those microtubules become fluorescent, and then you could see over time there's that dissipation of fluorescence. And again, if you have a highly dynamic structure, then you could see that that dissipation occurs very fast. And if you have a more stable structure, then the dissipation is much slower. So with these types of data, then what we could do is we could analyze it and have a decay curve. The same as we did for FRAP, where you have a recovery curve. Here we have a decay curve for this particular cell that I show here. And then what we could do is, depending on the data set that you have, you could either fit it to a single exponential decay or to a double exponential decay. And what that allows us is that we can then calculate our half time and see how fast we have this dissipation of fluorescence within that structure. And for another cell much later, or that could have a more stabilized structure, then you could see that the decay curve looks different from the cell I just showed previously, where you have not as much decay, and then it kind of plateaus and then stays constant. And again, you could fit it to a single exponential or to a double exponential, depending on the population that you're studying, or in our case, depending if we have two populations of dynamic and stabilized microtubules. So right now we're working on quantifying all this data and seeing how the dynamics of the mid-zone is changing as the cell progresses in anaphase, in addition to knocking down some candidate proteins that might be playing a role in how dynamic the structure is, and seeing how that impacts the dynamics once we don't have the protein present. And with that, I would like to say thank you for everyone who's here, and I'll take any questions.
Thanks, Carline. That was really cool. And Pat, very cool work. I always love seeing when Carline's on the microscope doing the photo activation. So awesome to watch in real time. So we're doing pretty good on time. So if anyone has any questions for Pat or Carline, I think you could just unmute yourself and ask it, or put it in the chat, or at the end of Joe's talk.
Change that.
Where's your chat?
Hi, I have a question. During the recovery after photo bleaching, what's happening during the recovery? You showed like there was was it like the interchange?
Yeah, so for that particular, so in that particular example, and I'm talking about the experiments that we did within the spindle, right? So for FRAP, we were actually seeing the exchange of tubulin dimers as the microtubule is growing. So what happens is, because you have the spindle is completely fluorescent, green fluorescent in this case. Once you bleach it, then those become you bleach it and it goes in. So you can't see it anymore. And then what happens, what you're seeing is that the bleached tubulin dimers are actually being exchanged with unbleached 2 mm diamonds, and that's how you see that recovery curve for us.
Oh, okay.
Yeah. And then so if you want to see a protein, how dynamic a protein is, then you could see also the exchange of that protein binding and unbinding, and the exchange of proteins that you bleached and proteins that you didn't bleach. And that's what the recovery curve is showing you. Yeah.
Oh, okay. Got it. Thank you.
I have a quick question as well.
Yeah.
Great presentation, y'all. This was awesome. I had a question about sort of the limits of detecting like really slow dynamics, and whether, I mean, I understand the sort of the exchange of non-bleached molecules for the bleached molecules. So theoretically, even slower dynamic processes, you could still get that curve. But is there any sort of limit on the technique in terms of measuring like slower dynamic processes? I would assume no, but I just yeah, I was just asking if that can be a technique that's used for things that happen over, you know, like a long period of time.
Right. So Pat might know more, but so of course, like I would assume that if you have a structure that is not as dynamic, then that recovery curve might look a little different compared to a dynamic. The other thing that could happen is that if you have a immobile fraction, right, there's no dynamics happening, then you won't be able to capture that, right? But in terms of how dynamic, I guess you would have to do it to try it to see how dynamic unless you know how dynamic the structure or how the protein is. But that's, I don't know if that answers your question, but I think that the other thing that you could keep in mind is also how you're collecting the data. So thinking about a dynamic structure, right? You might be collecting the data too fast where you're not able to actually observe how dynamic that structure is, because you're just, it's happening so fast and you're collecting it so fast, so much faster than when that dynamic is happening, which I guess wouldn't happen if it's not as dynamic, because you would catch it. But that's one thing that you keep in mind is actually how you're collecting that data, depending on how dynamic or non-dynamic that structure or protein is.
And for that opposite end, for like the really fast dynamics, are you just adjusting like the rate at which you're in your ND acquisition thing?
Yes.
Yeah.
Right.
Okay.
So
yeah.
the movie that I showed, the two movies that I showed previously, and I could go back to it, right? So I'm collecting at a much faster rate here compared to here, just for the simple fact that, as you could see, the fluorescence dissipation here is happening at a much faster rate than here. So yeah, so depending on how dynamic the structure of the protein that you're trying to study is, then you could adjust for that interval of how you're collecting the data. Yeah.
I mean, I just want to add, as long as you can keep your sample on the scope and turn on perfect focus, you could have a long interval between taking images. And so you could probably watch a recovery over an hour or a couple hours, as long as you perfect focus it and your cell is happy. I think it would be fine.
Cool. That's awesome. Thank you.
I think maybe the one other consideration, which I didn't hear, was actually photo bleaching of the photoactivated molecules, spacing out your acquisitions far enough that you're limiting that the destructive nature of the process. So I think we should move on to Joe at this point. So thank you, Carline and Pat. And if anyone has more questions for them, let's save it till the end. And Joe works with Nikon, and he's going to talk a little bit about single molecule localization. Thanks, Joe.
Thanks, Jim. Let me just share my screen here. All right. Well, thank you, Jim, for inviting me to. Can everybody see this?
We don't see your screen. We just see you right now.
Oh, maybe I need to. How about that?
Yes.
All right. So thanks, Jim, for inviting me. Today, I want to talk about storm imaging. And you guys have access, actually, to a beautiful storm system. And I know some of you are interested in using it. But sometimes people tend to be a little intimidated by storm. It's a little bit different of a technique than other types of light microscopy. And so hopefully today, after I talk a little bit about what it is and how it works, maybe you'll feel a little bit more comfortable about using it and have a better understanding of it so you can determine if it's right for your research, because it's really powerful, but it's not necessarily a tool for everybody. It really depends on what you're looking at. So any of you who study subcellular structures, like in the previous talk, right? We know that there's a major limitation with light microscopy, like traditional light microscopy, resolution limitation. And a lot of times you can actually see the structure that you're interested in, but you can't necessarily resolve it, meaning that you might be able to confirm that it's there. You might be able to learn some things about it, but to be able to accurately be able to say what the size of the object is or how many of the objects are there is not necessarily reliable. So for example, here we have actin, which are about 10 nanometers across. And we know in this wide field fluorescence image that these filaments are present, but we can't be 100% sure about how many there are or how big they are. So why is this? And we need to get into a little bit of how images are formed so that we can understand how storm works. The reason why we have this resolution limit is because in traditional light microscopy, you have this thing called diffraction limitation. Basically, images are formed by these overlapping things called point spread functions. And the size of these point spread functions is determined by the wavelength that you're imaging and the numerical aperture of the lens. Basically, when you have a fluorescence image, what you have are a bunch of these spots. And if we presume that each of these spots is light is a molecule, the closer that any of these two molecules are together, the harder it is to be able to distinguish them. And below a certain point, which is roughly about 200 nanometers, it's basically impossible. And this is just a limitation of the optics and physics. But obviously we have something called super resolution, and we call it super resolution because it allows us to break this limit. So how do we do that? Well, there's a bunch of different ways, but I'm going to talk about one way, which is localizing where those points of light came from. So with a few assumptions, you can accurately find the center of a diffuse spot. So we have a bunch of diffuse spots here. I guess my slide is a little messed up. That is a terrible estimation of where these spots are. I promise you we can do better than that.
I've seen it do better than that.
Yeah. So anyway, so basically what storm is, it's a localization based technique. So if you are able to detect a spot, then you can basically do like a Gaussian fit and determine with varying degrees of accuracy where the center of that spot is. So that's where storm comes in. It essentially utilizes images of these single diffraction spots so that you can determine where the point source of light is. Problem with conventional fluorescence, and the reason why we can't do this with conventional fluorescence is because, like in the previous talk where we were looking at these wide field or maybe the confocal images, you're essentially illuminating all of the fluorescent molecules in the sample. And so you have a bunch of these spots that are all over the place that form the image. And it's really hard to be able to determine what spots are where. So we need to figure out a way to reduce the number of spots we see at any given time. We call this emitter isolation. So basically, if you have a way where you can take a bunch of frames, kind of like a time series image, but instead of the entire sample being illuminated at any given time or fluorescing at any given time, you only have sparsely fluorescing molecules. So in this example, you see here, at any given frame, we only have a few, and because we only have a few, it's sparse. They're not overlapping for the most part, and so we can easily fit something to these point spread functions and determine where the source of light is. Essentially, what we're going to do with Storm is we're going to collect a ton of these, and then we're going to stack them together. We're going to basically determine where the spots are in every frame, and then you can put it together and it builds a image of what you're trying to look at. So how do we create this sparse excitation? Well, it's something called photoswitching. Basically, like if you're familiar with other fluorescent molecules, you're essentially exciting the molecule. It puts it into an excited state. And then when it drops from that state, you emit a photon. Now, there's actually another state called a dark state. And this dark state is a state where it doesn't fluoresce. And it can basically go from this state to the ground state directly without causing fluorescence. And basically the way that we do this is we use thiols and we use high laser power and we use special fluorescent molecules that I'm going to talk about in a little bit, and a special buffer that makes it more likely that the molecule will go into this dark state. So now we're exciting it like we normally would, but a lot of those molecules are going into this dark state. But they're not always going into the dark state. Occasionally, you'll have one that's excited and it will release a photon. And then in that frame, at that spot, you'll see a signal. And so if you have most of your fluorescent molecules in a dark state, and every once in a while, some of them do fluoresce, that's how we get that sparse emitter isolation that I talked about. Now, today I'm, mostly talking about Storm, but PALM is a similar process, except you are using photoconvertible, photoactivatable proteins or labels rather than photoswitchable labels. But the concept is the same. So, how do you build an image? Well, like I said before, we're actually going to take many conventional images over time, and then we're going to do the statistical analysis to determine where all the points are, and then you use that to create the final image, which gives you a resolution typically between 20 and 30 nanometers of resolution. So it's pretty good. And actually, because it's so good, and because of the way the image is created, you have to think about the image a little bit. differently than you would with normal microscopy. So how do we make sense of what we see? Basically, these storm images, they're not really images. They're more like scatter plots. And basically, what you have are points that denote the location of a detected molecule. And you might notice, it might be hard to see here, but you'll notice that some of the spots are different sizes. Or if you've ever looked at a storm image, you'll notice that the spots are different sizes. So what does that mean? Well, as with any kind of fitting function where you're trying to estimate the location of something, you're going to have a certain level of precision. And it's the same case with storm. So basically, the more photons you have from a location, the better the software is at being able to determine where that light came from. And so it denotes that location with a smaller spot. So you'll have spots of a different size. The spots that are smaller are the ones that are more accurately estimated where that light came from versus the spots that are bigger. So that's how storm works. Just to recap, basically, you're taking normal fluorescent images, but you're looking at a sample that is labeled and treated in a way that puts most of those fluorescent molecules into a dark state. Occasionally, those fluorescent molecules will fluoresce sporadically. We can use that sparse image to be able to determine where the light is coming from any given molecule, do that over time, and then be able to build an image, a scatter plot image of where these molecules are located. But there's some considerations, like I said. It's a little bit different than regular wide field fluorescence because you have to make certain choices and the preparation is a little bit more involved. So one thing that you have to consider is what probe you use. Now, there are different things to consider. There's a lot of literature out there that of people who have tested all different types of things. And so I definitely recommend that if you are to look through the literature and see what kind of molecules people have used in your types of samples. This is just a list of some of the ones that are out there. Things that you want to consider, you want something with high photon number because the more photons, the better your estimation is going to be in localizing your molecule. You want something that is going to have a lot of density within a location. And you want something that's going to be stable at high laser powers. Because remember, we're using high laser powers to help put it into a dark state. But that high laser power can also bleach these molecules as well. So you want something that isn't going to bleach easily. And sorry, I missed, I had one last thing there. Low duty cycle. Basically, that is how fast it switches states, right? So you don't want something that's constantly switching and emitting because then you're not going to have enough sparseness. We talked about the resolution of storm, 20 to 30 nanometers. At this point, we're at such a small scale that the dye that you use actually matters. And so you can see here we have 3 different dyes, and depending on what dye you use will determine what you can see, because if the dyes are particularly big, or for example, the label size is bigger than the resolution of the technique, such as this case where you have a secondary antibody conjugated to the label antibody of interest, creating this long chain that is basically the same length of what we can resolve. Now the location of the point source of light is going to be where the light came from, not necessarily where the protein is that you're labeling. This is known as linkage error. And, you know, there are ways of dealing with this. For example, you can use shorter secondary antibody or nanobodies. You know, you can do things with like direct conjugation to proteins, but this usually reduces the labeling density. So, you know, you're kind of gaining one thing for the other. And it really depends on your question, and it really depends on how good you really need to localize these things. Of course, you can use fluorophores that are close to the protein of interest, but this may lead to low photon yield. So that's labels and choosing labels, which labels you choose, and both in terms of their properties and in terms of their like photon emitting properties and also their size properties. Another thing that you need to consider is the imaging buffer. And the imaging buffer is important because it's cooked up in such a way and it contains chemicals that help prevent photobleaching, such as these oxygen scavengers. And it also stabilizes that dark state, like I mentioned before, typically with thiols. So MEA or BME. Jim can definitely probably help you with different storm buffers and what's worked for him in the past. Definitely need to, make sure that you look through the literature, that you use fresh buffer. And I'm going to go through some tips here. So, very rarely, storm is one of those things where when you get it to work, it's amazing, but you don't get discouraged if it doesn't work the first time, because there are a lot of moving parts here and everything needs to work properly in order to be able to produce a nice, beautiful image. So don't be afraid to play around, try different antibodies from different sources, see what works best for your sample. You want to make sure that you optimize the fixing of your sample. You want to really preserve the structure. You want to have really thin sections so that you can reduce background, and you want to maybe try different amounts of your primary antibody and see what works best. Blocking serum, we can talk about that. You want to make sure that you wash at every step, and you want to do a post-staining fixation and clean again. Try different buffers, and make sure that you have everything in them. If your buffer is old, there's been many times where we've tried to do storm imaging and, someone brings us a buffer and we're not getting that blinking. And it turns out, well, the buffer is a few weeks old and, who knows, maybe it's sat out or something like that. So just make sure that you're doing things fresh. Now, when it actually comes to doing it, and I'm almost done here, basically the controls are pretty straightforward. You have your choice of doing 2D or 3D storm. You basically have the laser lines and the laser powers. You have the number of frames that you want to capture, where you want to save it, and then there's a summary of the experiment. This is the main window that you're going to work with. There are some other windows to do settings and stuff like that. And if you were to want to do storm and be trained on it, you'll be shown those. And then when you get the images, then you have to do the analysis workflow, which is basically just setting the boundaries for how you want to determine what you consider a spot and not. And so there's a little bit of trial and error in this as well. And that's basically, once you do that, that's what produces the storm image. And so there are a couple things that I didn't really talk about, but if you have any questions, you know, we can get into it a little bit, such as multicolor storm, 3D storm I did touch on. And then this third picture here is actually changing the angle of the beam and doing storm with turf to kind of reduce that background. So with that, thank you. And if you have any questions, feel free to put them in the chat. I'll pull the chat up here.
Thanks, Joe. That was really awesome. Yeah, feel free to either raise your hand or just unmute yourself. I don't think there's too many folks here. So, okay, we have a question in the chat. Can it only be done in slices?
Well, basically, you're only able to look at something that is right on the other side of the cover slip. And so slices, actually, you know, really you want like tissue culture is like something really thin. You can do slices, but once you start getting too thick, you risk having a lot of background. It's certainly been done. There's no doubt about that. And people do it even with live cells and with live tissue as well. But, you know, there's going to be a little bit of work to get it to work. And, you know, I think Jim can attest to that, right?
I'm not going to comment. Yes.
Not going to comment. Okay. But certainly, yeah, certainly if it's your first time doing it, I would definitely try first having thin slices, fixing your tissue, making sure that you have antibodies that work and that you're able to do it the basic way before you try and do it in a live, like thick slice or something like that.
Yeah, I totally agree. Tom, do you have a question? No, we can't hear you for some reason.
Rena?
A little bit.
Okay, I'll speak in the microphone.
That's better.
Okay, so when you say thin, typically, you know, we've got two levels that we want to look at, even on cell culture on slide. We have what's directly below the membrane or above the membrane. And then we have the nuclei, which is going to be about 5 micron on top of that. So can you reach that far? 5 micron?
Probably
5 micron
not.
For the nuclei. Can you reach up into
5 microns?
Yeah.
And I think that's pretty far. You know, typically we're doing, you know, most of the time when you get a really good storm image, you're doing turf. And so you're looking, you know, hundreds of nanometers like above the cover slip. And so you start getting microns above, there's going to be a lot of background and it's going to be tough. Yeah. Jim, I mean, what's the furthest you've looked?
I mean, we've done a little bit of dirty turf. You know, if the background isn't too high, then we can kind of tilt the turf angle up a little bit to give us a little more distance. But the background has to be really low for that to work. It does.
And so that goes back to what I said before, which is really nailing down the preparation of your tissue, doing your washes, using the minimal amount of antibody as you can, making sure that you wash it between steps, those kind of things, all those things that would always make a better sample, but maybe aren't as important when you're using a technique that like confocal, for example, where you can get rid of that out of focus light.
So there's another question in the chat right now. Can primary neuronal cultures suffice?
Well, I've never personally looked at neuronal cultures with storm, but I don't see why not, as long as they're right on the cover slip. I think theoretically it should work. Yeah.
Yeah, I agree.
I don't want to make any promises, but yeah. No, but I think so. Yeah.
Were there any other questions from anyone in the audience? If not, I have a quick question, more like logistics. So when people are, maybe you could just say a few words about 3-color storm. What's the best way to attack that? Do you image the longest wavelength one first? Do you intersperse them?
Yeah, I mean, I've seen success with both ways of doing it. And I think it really depends on the fluorophores. I know that's kind of a cop out and the sample, but typically I do the longest wavelength first. I do that with a lot of imaging, honestly, just because theoretically that's the better way to do it. But if you, obviously, if you're doing something really complicated, like a living sample or something that's moving, then you have to intersperse them, right? So it really depends on your question and what you're asking. But if you have a really stable sample, then I would start with the highest wavelength and then collect your sequences one after the other.
Awesome.
But we'll try different things next calendar year and see.
Yeah. So we're calling basically the winter is the stormy winter. So it's a little inside joke. So we're going to be holding a few workshops and on-campus events and whatnot. So keep an eye out for all the normal marketing channels through ISLES to get involved with those. And Joe will be on campus at some point once we figure out some schedules. So with that, I want to thank Joe for a great talk, as well as Carline and Pat. Thanks for showing us your science. It was really cool.
Yeah.
And thanks, Joe.
Thank you.
Thank you, Lisa.
Great. Thank you, Jim. Thank you, Joe, Pat, and Carline. And thank you all for attending today's seminar. Our next and last seminar of this semester is Tuesday, November 30th, which will be hosting the UMass Glassblowing Lab with Director Sally Prash. She will give a short tour of the glassblowing lab, discuss how and why glass is used in scientific research, and give a short demonstration of how one-of-a-kind scientific glass is made from start to finish. Hope to see you there. Goodbye, everybody.
Sounds kind of cool, actually.